Hello students, today we are discussing about the second part of our journey from DNA to chromosome. So you can see on this paper, today's topic is second part is chromatin. See, the word chromatin means chroma means color and tin means filamentous structure. The structure, which is the after structure of supercoiling of DNA and before structure of the chromosome, or can say the middle stage in this journey, is chromatin. Chromatin is a complex of DNA and protein found in eukaryotic cells, means they are absent in prokaryotic cells, packaging of long DNA into more compact form called chromosome. After that, these chromatins will get supercoiled and form the last structure of our journey is chromosome. You can see proteins, those are included to make this supercoiling, are known as basic histone proteins. Histone proteins are only found in eukaryotic cells or well defined cells, means they are lacking in prokaryotic cells. There are basically five types of histone proteins. First is H1, or denoted as H1, comma H2A, comma H2B, comma H3 and H4. All type of histone proteins are held together to form an octamer stage known as histone octamer. In this octamer, there are four type of histone proteins in central part, but these four types of histone proteins are in double molecular structure, means 4 into 2 is equal to 8 molecules of 4 types of histone proteins. You can see left side of this diagram H2A, H2B, H3 and H4, and their back side another molecule is also attached. To bind this structure, or to stable this structure, last histone protein, or can say first histone protein, the H1, stick with them and pack this structure. Next point is that the coiling of DNA around this histone protein is in 1.7 turns, means the complete 1 turn or complete single turn, and the second turn is less than zero point 0.3. How many base pairs are present in octamer? There are 147 base pairs. Okay, the entire structure is known as nucleosome. You can see the central diagram, the DNA, and then addition of histone protein, which is drawn in green circular octamer diagram, and these histone protein binds with DNA to form a bead like structure known as beads on a string, and then they are superly coiled to form chromosome. So guys, the exact diameter of nucleosome is 30 nanometer. You see, if there is no histone proteins in prokaryotic cell, then how DNA gets supercoiled? So answer is that supercoiling of DNA in prokaryotic cells is absent or absolutely absent because they lack histone protein, or can say the coiling protein, which is necessary to coil the DNA into chromatin and further chromosome. In prokaryotics, or in prokaryotic cells there is another structure which is exactly look like a chromatin known as geno4. See how many types of chromatin found in eukaryotic cells. So on the basis of their activity, their color, their density and their position in chromosome, there are two types of chromatin. The first is euchromatin and the second is heterochromatin. Euchromatin is transcriptionally active, means this chromatin can prepare different types of proteins and is functional. But heterochromatin is turned off or non-functional in human body or any other animal's body. Euchromatin is lightly stained chromatin, means if we can stain the chromatin, so we need some staining agents such as acetocarmine, hematoxylin. These chemicals could not stain properly this chromatin, that's why it look like very light in shade. But heterochromatin, because of its density, heterochromatin looks very dark under the microscope. Euchromatin is comparatively thin as compared to heterochromatin because heterochromatin is more supercoiled or more dense structure. See, euchromatin central in position in a chromosome and peripheral in position is heterochromatin. You can say some examples of euchromatin proteins are housekeeping genes. What are housekeeping genes? The genes, those are responsible for day to day activities of our entire body. So these are housekeeping genes. And the example of heterochromatin into the neutrophiles are bar body, or can say an extra X chromosome which is converted into heterochromatin and get re again or so many times converted into X chromosome while oogenesis. Next point is that the word gene was given by William Johnson, but before him genes are or genes were called as factors. The word or the term factor was given by Mendel or Gregor John Mendel. So question is that what are genes? You can see the diagram. DNA has two strands. If you can cut this DNA into several segments or into several pieces, so each piece or each segment is itself a gene. So the definition is that either small or long segments of DNA, those are either functional or non-functional. You can see there is approx 80% of our DNA is non-functional. At least 10 to 20% is functional for our body activities. Next, avian RBC means the RBCs of birds and sperms has more compact chromatin than other eukaryotic cells. Okay, hope you guys understand about this topic known as chromatin. Thank you.